our discussion, let us start with a prayer. Please bow your head. Let's pray. Before we move on to our discussion, we will first have an activity. You have 15 seconds to complete the activity. You will find the words that is related in transcription and RNA processing. Are you ready? If so, let's start. Now, let's see if your answers are correct. Well, you get the correct answer. Now, let's move on to our discussion. Our lesson for today is all about transcription and RNA processing.
As we go along to our new lesson, I have a question, class. What is transcription and RNA processing? Anyone can give an idea? Okay, Mr. Virgilio, you're raising your hand. That's right. Very good. Transcription, it is a process in which a gene's DNA sequence is copied or transcribed to make an RNA molecule, while the RNA processing serves as a multitude of functions within cells. These functions are primarily involved in converting the genetic information contained in a cell's DNA into proteins and determine the cell's structures and functions. It is also the process by which RNA polymerase enzyme and other transcriptional proteins and enzymes use the template strand of DNA to synthesize a complementary RNA strand. So, when we say RNA polymerase, it is a multi-subunit enzyme that applies its activity in the catalyzation of this transcription process of RNA synthesis from DNA template. It is also the specifically build an RNA strand in the 5' to 3' direction, adding each new nucleotide to the 3' end of the strand. The RNA polymerase locally opens the double-stranded DNA so that one strand of the exposed nucleotides can be used as a template for the synthesis of RNA. This process is what we call a transcription. Structural comparison between DNA and RNA. So when you say DNA, it is the responsible for storing and transferring genetic information. These molecules are typically long and double-stranded. It contains sugar, deoxyribose, and thymine, while the RNA are much shorter and typically single-stranded, so it can fold upon itself, forming a three-dimensional structures. Each RNA strand has the same basic structure, composed of nitrogenous bases covalently bound to the sugar phosphate. These are the adenine, cytosine, uracil, and guanine, and they play a major role as building blocks of the nucleic acid. RNA is the product of transcription, which can be encountered in the form of the three major types, which is the messenger RNA, ribosomal RNA, and lastly is the transferred RNA. Messenger RNA or mRNA, it is created during the process of transcription. It is a single-stranded RNA molecule that is complementary to one of the DNA strands of a gene. Specifically, mRNA carries the protein blueprint from a cell's DNA to its ribosomes, which are the machines that drive protein synthesis. The ribosomal RNA or the rRNA is located in the cytoplasm of a cell, where ribosomes are found. These are the molecules in the cells that form of the protein synthesizing organelle known as a ribosome, and that is exported to the cytoplasm to help translate the information in the messenger RNA. Lastly is the transferred RNA or the tRNA. It is a highly folded structure that holds an amino acid on one end and has what is called the anticodon. It is an adaptor molecule composed of RNA, typically 76 to 90 nucleotides in length, and serves as the physical link between the mRNA and the amino acid sequence of protein.
Transcription in prokaryotes, catalyzed by a single RNA polymerase, is the process in which messenger RNA transcripts or genetic materials in prokaryotes are produced to be translated for the production of proteins. It occurs in the cytoplasm alongside translation. It doesn't require any proteins or other factors for the initiation of transcription. Transcription in eukaryotes. It's catalyzed by 3 different RNA polymerases: the tRNA, the mRNA, and the rRNA. It's the collaboration process that eukaryotic cells use to copy genetic information stored in DNA into units of transportable complementary RNA replica. It occurs within the nucleus, where DNA is packed into nucleosomes and higher order chromatin structures, and it requires proteins known as transcription factors for the initiation. Here are the 3 steps of transcription. First, we have the initiation. It is the beginning of a transcription. It occurs when the enzyme's RNA polymerase binds to a region of a gene called the promoter. Each gene has its own promoter. Once bound, the RNA polymerase separates the DNA strand, providing the single-stranded template needed for transcription. The second one is the elongation. It is the process through which nucleotides are added to the growing RNA chain. During the elongation, the ribosomes continue to translate each codon in turn. Each corresponding amino acid is added to the growing chain and linked via a bond called peptide bond. Elongation continues until all of the codons are read. The last one is the termination. It is the ending process of transcription and occurs when RNA polymerase crosses a stop sequence in a gene. When the mRNA strand is complete, it will detach from DNA. Since there are no tRNA molecules that can recognize these codons, the ribosomes recognize the translation is complete. The new protein is released and the translation complex comes apart.
Now, let's have the generalization of our topic. Class, what is transcription? Anyone? Very good. Transcription is the first step in gene expression. It involves copying genes' DNA sequence to make an RNA molecule. How about the RNA? Anyone can give me an idea? Very good. Ribonucleic acid or RNA is a molecule similar to DNA. An RNA strand has a backbone made of alternating sugar ribose. Structural comparison of DNA and RNA. What is DNA? Anyone? Very good. DNA is stable under alkaline condition. Molecules are typically long and double-stranded. It contains slightly different sugar, deoxyribose, and thymine. How about the RNA? Anyone? Very good. RNA molecules are much shorter and are typically single-stranded, and it can fold upon itself, forming a three-dimensional structure. It contains sugar ribose and has nucleobase uracil. RNA polymerase is a multi-subunit enzyme which applies its activity in the catalyzation of the transcription process of RNA synthesized from DNA template. Specifically builds an RNA strand in the 5 to 3 direction, adding each new nucleotide to the tree end of the strand. Anyone can give me the 3 major types of cellular RNA? Very good. The 3 major types of RNA is the messenger RNA, ribosomal RNA, and the transfer RNA. Messenger RNA is the first step in making the protein from a DNA blueprint, made up of nucleotides, non-coding region. The ribosomal RNA is having an important role in translation, 3 sites called A, P, and E. Transfer RNA is the decoding device used in protein synthesis. It is a highly folded structure that holds an amino acid on one end and has what is called the anticodon. Class, what are the 3 steps in transcription? Very good. The 3 steps of transcription is the initiation. It's the beginning of the transcription. It occurs when the incised RNA polymerase binds to a region of the gene called the promoter. Next is the elongination. It is the process through which nucleotides are added to the growing RNA shape. During the elongination, the ribose continues to translate each codon in turn. The next one is the termination. It is the ending of the transcription and occurs when RNA polymerase crosses a stop sequence in the gene. Transcription in prokaryotes catalyzed by single RNA polymerase. Transcription in eukaryotes catalyzed by 3 different RNA polymerase is the elaborate process that eukaryote cells use to copy genetic information stored in DNA into units of transportable complementary RNA replica. Occurs within the nucleus where DNA is packaged into nucleosomes and higher order chromatin structures. Requires proteins known as transcription factor for the initiation. And that is the end of our discussion. Now we will have an activity. If you really listen to the discussion, you will be able to answer the question immediately. This is 10 question, multiple choice. You choose the correct answer to the question. You have 30 seconds to each question to answer. Are you ready? If so, let's start.